In this lesson, we'll be talking about the operation of a bright field microscope or light microscopy. So in this course, we'll be using bright field microscopes, which use a combination of glass lenses to magnify an image and then light to view the specimen. With the type of microscopes that we work with in our course, we'll primarily be looking at things like bacteria, fungi, and protozoa. Here we have a few images of some standard microscopes that are utilized in labs such as our own. So we kind of look at these various different places on the microscope. Microscopes will typically have a base, and in the base or the bottom of the microscope, you are going to have the light source, which we see here, which is also called the illuminator. The light source or the illuminator's job is to really project light upward. So the light source here will contain a light bulb, and then the illuminator is going to project that light upward. From there, we get to the condenser, which is this entire structure here underneath the stage. The condenser's job is to do kind of what its name says. It's meant to kind of condense or collect and focus the light. What typically happens is the light that leaves the light source through the illuminator is going to project upward like a flashlight, so a lot of it's going to be lost due to scatter. So what the condenser does is it kind of collects and focuses all that light to shine it straight to the specimen, so we kind of reduce the scatter that we would see here. Within the condenser, we have the diaphragm control here. And this is meant to really control kind of how much light escapes that condenser. So this can be adjusted depending on contrast if you want more or less. Here we have our focusing dials, so over here and then over here as well. Our focusing dials do exactly what their name says. They are meant to focus the image. There's the fine focus and then the coarse focus. The fine focus is the smaller dial here and the smaller dial over here. And then the coarse focus is the larger dial. Both of these are meant to move the stage up and down to bring the specimen into focus. The coarse focus is going to move the stage in very large increments up and down to bring the image into focus, whereas the fine focus, it generally is going to move the stage in very small increments for focusing. Typically, we will use the coarse focus only with the smallest objective, which is the 4x objective, and then under all other objectives we are going to use the fine focus. Then we have the stage. The stage is here with little stage clips as well that you can see on this microscope, and that is meant to hold the specimen that's on the slide. We do also have stage controls, which are located over here and over here on this microscope. The stage controls basically just move the stage forward and backward and to the left and right so that you can center your specimen underneath the objective lenses. So then we have our objective lenses, which are here in this image and then over here. So the objective lenses are meant to magnify the image, though they do it in varying degrees. So typically, when you're working with a microscope, you start out with the smallest objective lens, which magnifies by 4, and then you gradually progress upward through all the other objective lenses. But they do serve to magnify the image, and you can vary the degree of magnification depending on which one you have clicked into place. And what rotates through here is this revolving nose piece, which you can kind of rotate through to adjust which objective you want into place. Both of these microscopes also have a carrying arm that can be used obviously to carry the microscope from one place to another. Anytime you carry a microscope, you should also support the base of the microscope with another hand, as they are quite heavy. And then up here we have the eyepiece here and here, which has the ocular lens built into it, which is also a magnifying lens that allows us to view the virtual image, which is perceived by our brain. On this microscope, we have one ocular lens. On this microscope, we have two. In our labs, it's common that we will use a microscope with two ocular lenses. In regards to microscopy, there are three primary principles: magnification, resolution, and contrast. Magnification is the ability to increase the size of an image or make it larger. So this is typically done with the objective lenses, which magnify by varying degrees, and then the ocular lens. Then there's resolution. Resolution is basically the ability to see something clearly. So it's the ability to see something with fine detail. Because we can magnify something all we want, but if we magnify it and it's blurry, it doesn't really do us much good. So we want to magnify in a way that we also achieve resolution, which can be challenging at magnification such as 1000x, and we'll talk here in a second how we can overcome that challenge. And then the last is contrast. Contrast is the ability to see cells against their background. So naturally, bacteria really don't have a color. They're really kind of like a light gray to tannish color. And if you tried to see them against a white background, you wouldn't be able to do it very well. So typically we will stain bacteria pink, purple, blue, green, whatever, in order to create that contrast against the white background of a microscope slide. Or even with this text on this page, it has good contrast because we have a light background and dark text. But if I would have chosen light pink text against this light background, obviously it wouldn't have had good contrast. So in regards to magnification, again, magnification is the ability to make a specimen appear larger under the microscope. So for us, we have two magnifying devices within our microscopes. So we have the objective lenses and then the ocular lens. So the objective lenses, and again, those are the ones that we rotate through here on the revolving nose piece, they are going to vary in their degree of magnification. So you always start out with the smallest, which magnifies by 4. This is called a scanning objective, and it just allows you to kind of scan the slide to see the area that you want to focus in on, which is usually where you see some type of color contrast since we stain the cells. After you find the specimen with the 4x objective, then you progress to the 10x, which is the low power objective, which gets you more magnification and a little bit more detail. Then you get to the 40x objective, which we call the high power objective. This is also called the high dry objective sometimes because it's the highest you can go without using oil immersion. But again, it just gets you more magnification, another level of detail for the specimen. And then we have the highest power objective for our microscope, which is the 100x oil immersion objective. So this is the objective where you have to have a layer of oil on the microscope slide in order for this objective to work the way it's supposed to and get the highest level of magnification with the best resolution. So then we have the ocular lens. So the ocular lens, if you remember, is built into the eyepiece up here. So the ocular lens, you cannot change the intensity of it. The ocular lens is set for a standard 10 in our microscopes. There are some other microscopes that are different, but in our microscopes it's set to a standard 10, so you don't have the power to manipulate this. But this will magnify the image further by 10 and create that image that we perceive by our brain called the virtual image. A common question you might ask yourself is how many times has this image been magnified so that I can visually see it under the microscope? And that is what we refer to as total magnification, which is very easy. All you have to do is take the power of the objective lens, whatever one you have clicked into place, the 4, the 10, the 40, or the 100, and then multiply it by the power of the ocular lens, which is always a standard 10. So if you're looking at a specimen under the low power objective, you would take 10, which is the power of the low power objective, and then multiply that by another 10 from the ocular lens, and that gets you a total magnification of 100x. Most of the time in microbiology, because the microorganisms that we work with are so small, in order to see them clearly, you're going to have to view them under oil immersion, which magnifies by 100. and then if you account for the 10x from the ocular lens, that means the total magnification is 1000x. Now these numbers may change on quizzes or exams, but just keep in mind the formula. You just take the power of the objective lens and then multiply it by the power of the ocular lens. Once you multiply those two numbers together, that gives you the total magnification, and then you just put an X on it for units of measurement for magnification. So resolution or resolving power is the basically ability to see an image clearly. It is the ability for a lens system to distinguish two points from one another, so to see two objects as two separate objects instead of one kind of big blurry unified object or image. And in micro, how we achieve resolution is by using the oil immersion lens and a drop of immersion oil. So typically when we use a microscope, what can happen under higher magnifications is the light can get refracted away. And we want as much of that light as possible to shine through the specimen to illuminate the image so that we can see it clearly. So in order to capture some of this light that's typically refracted away, what we do is we put a drop of immersion oil on the slide. That is going to capture all that excess light so that it goes straight through the specimen and then towards the objective lens in order for us to visualize the image more clearly. So it's all about collecting that excess light via the immersion oil and the oil immersion lens in order to achieve resolution or a clear image. With our microscopes, we are limited in what we can see. So with the standard microscopes that we have, the resolving power is about 0.2 micrometers or bigger, or 200 nanometers. So this would allow you to see animal cells, including human cells, protozoa, algae, fungi, and even bacteria, although you wouldn't see it in very great detail. So this is a standard microscope here, and this is the image that we would expect here of bacteria using a standard microscope. So these little tiny purple bacillus rods, those are all salmonella. Over here we have an electron microscope, which is not something that we have in our labs. Electron microscopes magnify at very high intensities, and this is what you would need in order to see a virus. So we are not going to see any viruses in our lab. We kind of max out the smallest thing that we can see, our bacteria. But with electron microscopes, you would be able to see viruses. And this kind of compares what salmonella would look like if you were looking at it with a standard microscope versus an electron microscope. And obviously, with that high of magnification, you would see a lot more detail. Adding immersion oil can be a little tricky. It's kind of new to microbiology. It's probably not something used in other bio courses and anatomy courses because it's not necessary when you're looking at larger cells like human cells. But in microbiology, we have to look at bacteria, which are very small and require a higher level of magnification. So you have to use the oil immersion lens and the immersion oil. So when we are using the immersion oil, what you don't want to do is add it at the very beginning and then drag all your objective lenses through it. You only want to add it once you're completely focused under the 40x objective. So you focus under the 40x objective, and then once that's completely focused, you rotate halfway to the 100x objective, so you kind of create this V. You drop the immersion oil in on the slide, just one drop, and then you can click the oil immersion lens into place. So really the only objective lens that should come in contact with the immersion oil is the oil immersion lens. You do not need to put it at the beginning and drag all the other objectives through it. I highly recommend that you watch this video here to kind of get experience of how to utilize a microscope. And then you definitely want to make sure you look at these tips on how to focus a microscope, whether you're doing it in person or virtually. These are kind of some standardized tips that will help you with focusing that image.